For our last lecture on building animals, we're going to look at a pattern that we've already talked about when we began the class, but we did it in kind of a very rudimentary form where we were looking at it as an example of an organizer and how if you transplanted or ablated a bit of cells that were acting as an organizer, you could impact the phenotype. So remember when we talked about how we could cut off what was called the apical ectodermal ridge, or AER, which is the very tip of a developing limb bud in a vertebrate. And if we cut that off, that we wouldn't develop the distal portions of the limb. Or if we transplanted it, we'd get a duplication of those proximal to distal patterns. And so we basically get two kind of fused limbs growing out of the same stump, out of the same proximal portion of that limb. So limb development has some really interesting patterns, and it get very, very complex. We're going to do it in a little bit more complexity than we've done heretofore, but nothing really, really scary, really dramatic. So here are some of the patterns that we're going to be just looking at. This is just like a big picture. Just many of these are going to be very familiar to you. So the positioning of the limb along the anterior to posterior axis. So basically, where do limbs grow? And realize that's important because if we start growing forelimbs that are too far down, right, coming out of the abdomen, that's going to be bad. They need to be patterned to be growing right where the pectoral girdle is. So basically, how do we define where that pectoral girdle is for the forelimbs? How do we define where the pelvic girdle is and where the pelvis is going to develop and therefore hind limbs? And so that's a really important thing, and that varies somewhat across different vertebrates. And then once we establish that, then we need to do the initial outgrowth of the limb bud, which is the same gene. This is one of those deep homology genes, distalis. The same gene that begins to start that growth development. But then we need to start recruiting and signaling all these different limb organizers. We need to do the proximal to distal axis, right? So we need to start patterning where the shoulder joint or the hip joint is going to be, where the elbow joint, right, those are kind of medial parts of the limb, the elbow or the knee in the hind limb, and then of course the most distal elements. And we're going to look at some specific patternings of these most distal elements and then how we get forelimbs and hind limbs to be different from one another. So we're going to touch on all of these, but not in a lot of detail or complexity. Okay, so let's look at number one. Where do we grow the forelimbs? Where do we grow the hind limbs? Now realize this varies quite dramatically, particularly if we count somites. Now the skeleton is a really good model to look at for somites because we have a whole bunch of them that are integrated into the head, and that gets complex, but there's some similarities between heads and skulls across all vertebrates. But then in the neck, it's pretty easy. So notice that this swan has many different somites that all become these individual vertebrae in the neck. Whereas with a frog, we basically just have one little neck somite that connects so you can rotate the head a little bit. But there's just one somite between the head and where the forelimbs are going to go, where here we have dozens of them. So that can be quite different. Now, this is regulated by one of the Hox genes. I don't care that you memorize exactly which Hox gene it is. Just know that one of the Hox genes. Where Hox C6 is expressed, that defines where we are going to have the pectoral girdle, or in other words, the forelimbs growing, okay? And that varies quite dramatically. The position varies distinctly across organisms. So if we look at a frog to a swan, that's really, really amazingly different. But in some groups, in particularly in mammals, it's very, very conserved. There are only a couple of species, I think some sloths, that have a different number. There are 7 cervical vertebrae, which are the neck vertebrae. Cervix is basically just means a narrow region. And so cervical vertebrae are the narrow region between the larger head and then the larger pectoral girdle, the shoulders. And so in mammals, there are only 7. And so I think, again, with 2 species of sloth being an exception to the rule, but almost all mammals have 7 vertebrae. So it's kind of a cool pattern. We'll look at it again, come back to it here in just a little bit. Okay. So then once that's established, once we have established where the forelimbs are going to grow, which is Hox C6, once we have established where the hind limbs are going to grow, different Hox genes, but also Hox genes that establish where those hind limbs are, then we need to begin growing it. And we've already established this. We won't get into much. We'll do a little bit more detail, but realize that AER is the primary signal that says, hey, here's the distal side of the limb. Here is the proximal side far away from where the AER is. And if we mess with it, we end up with mutants, or we end up, if we graft on an extra one, we end up with duplicated versions. Fairly simple. okay? But once we have established this, realize this AER only establishes one of the axes, that there are other axes. So the dorsal to ventral axis is established by a whole different class of genes. And I'll notice this is kind of cool. The engrailed gene, which is a homolog of the engrailed gene that makes the segments, plays a different role here, but still somewhat conserved. Engrailed is one of the copies of engrailed, because there are actually more than one, but one of them in vertebrates established the the ventral pattern. The dorsal pattern and dorsal region is established by others, but it's part of that dorsal to ventral. So we have this same thing that we've already talked about with Drosophila, where we have secondary fields being established, but different genes. But some of the same genes pop up in slightly different roles. So Wnt is actually a paralog of wingless, which plays a role in wing patterning. Here it plays a role in establishing the anterior region of the developing limb. So we see the same sort of thing where we have the same axes, anterior to posterior, dorsal to ventral, proximal to distal, but in a smaller region set up by different groups of genes. Okay, so same basic pattern, but different genes playing roles in what the different parts are going to be. So let's look at some of these limb organizers. This is a really cool one because Hox genes kind of play a double role in the developing of the vertebrate limbs. They are part of the signaling process for where the limbs are going to grow, but they also play a role a little bit later downstream in determining what the different parts of the limbs are going to be. And this is particularly interesting for the most distal element, which is called the hand in humans, right? Or the claw or the paw, right? These most distal elements. So if you want to think about it as like the wrist and the phalanges, right? The fingers and the wrist, or the toes and the ankle, if you're looking at the hind limb, right? So, but this is really, really interesting because there's this really cool kind of folding over of Hox gene expression. So in the early limb bud, the proximal to distal elements for the most proximal part, the medial parts, those are all pretty basic. They basically start here in the developing limb bud and then go straight out. But when you get to the hand or the wrist and the hand, there's actually a folding over of this axis. So instead of just going all the way straight out to the tip and then fingers, wrist bones, and then elbow bones and so on all the way back to the shoulder, actually what happens is the shoulder, the elbow, the radius and ulna are all kind of patterned in that same, as you might expect, in that same axis from proximal to distal. But then the hand elements almost have this folded over. So it's like you go out here and then curve down to get the most distal element. So this kind of really unique pattern that is developed that made this complex distal part of the hand or the foot, if you will. And so this was determined originally by knocking out different Hox genes and then seeing what the deficiencies were. And so the phase 3 expression is this unique pattern going from what was originally the anterior to posterior part of this developing limb, but now has been reworked to become part of the proximal to distal element. And eventually there's apoptosis, these cells die out, and then the hand becomes kind of folded out to become the most distal element of the limb. So elements are still patterned in a proximal to distal pattern, but with this kind of folding over end pattern for the hand itself, which is kind of different and very distinct. And many of these genes do show some redundancy, which is not unexpected in the vertebrates because we have these many duplicated versions of the Hox genes which are involved here. There's some redundancy. So if you knock out one, you might not get a phenotype or only a very weak phenotype. If you knock out two orthologous copies of that gene, you might have a much stronger phenotype and be completely lacking or have major deformities in these distal elements if we knock out these genes that are responsible for patterning. So, take-home message. The anterior to posterior axes play a role in the patterning of where the hand or the foot is going to grow. And that's somewhat distinct because you expect that to be part of the proximal to distal patterning, but the anterior to posterior patterns play a role there also. And then there's some redundancy here. Okay, so number 4 is how do we get differentiation of hind limbs and forelimbs? Because in most of the higher vertebrates, they're quite different, right? Wings are different than legs in birds. Wings are different than legs in bats. Forelimbs are a little more similar, but still different and distinct from the hind limb in a horse or a cow. And in humans especially, there's quite a distinct one, a little bit more than other primates, right? Where there is distinction between forelimb and hind limbs, but legs are a little bit more similar in form and function, like in a spider monkey, than they are in a human, where there's quite distinct and their function is very, very distinct, right? Your arms do something very different as a human than your feet do, okay? Now, this is as you might expect, some topics that we have already talked about play a very, very important role here. So the pattern is thought to be that initially, in the earliest chordates, there was just one kind of long fin-like structure that was basically an appendage, but not as distinct as it is in modern vertebrates. And this is controlled, the expression of some downstream genes is controlled by engrailed. Later, there was a duplication of the gene that controlled this kind of one large long fin. And so this duplication allowed for redundancy initially, but then eventually a step to where we have a difference between the anterior appendage and the posterior appendage that are now controlled by different genes. Now, initially, in the earliest organisms, the forelimbs, or in this case they were fins, the forefins and the hind fins were very, very similar in both function and in the way that they looked. But later they began to diverge. And in one major group, the bony fish, this hind limb was essentially lost. And so in the bony fish, we essentially only ever grow the forelimbs. And these are called the pectoral fins, right? Which here in this group of bony fish, but a side group, are different, are here. So here are their pectoral fins, but these guys maintained their pelvic fins. Most bony fish lost the pelvic fins. These guys maintained them, and eventually a branch of this group of organisms became amphibians. Now, this is a living extant version of this. It used to be a much more diverse group. Today, they are only represented by the coelacanth, which was thought to be extinct until we kind of rediscovered it. Sometimes it's called a living fossil. And the lungfish, which we've known about for a long time. Lungfish have both pectoral fins and pelvic fins. And again, most fish have lost the pelvic fins, okay? But in this small subgroup, those became very important. They eventually developed into amphibians, and then the pelvic fins took on even more distinct function as hind limbs. But in all the early arthropods, we basically have this duplicated identity where the TBX and the TBX5 and the TBX4 both control appendage development, but they maintain their redundancy. So if you look at all of the early amphibians, and some modern ones too, like salamanders, there's a very, very similar phenotype between the forelimbs and the hind limbs. We still see much of this redundancy. But in later amphibians, like frogs, which are more recently evolved, and in many, many, many of the higher vertebrates, right, reptiles, mammals, there's a distinct differentiation between the forelimb and the hind limb. And so this is possible because of the duplication of this TBX4 and 5. This is the ancestor. It was the one gene. But the duplication of it allowed for differential control of the forelimb and the hind limb. So in this very simple beginning, we can get quite distinct modeling. And so here we have it in the chicken. TBX5 expression develops a wing. TBX4 expression develops a leg, and that's very, very different. So these originally the same gene, which now duplicated into different genes, control different downstream genes, and so we can get them. You can see this is somites here. So you can see where they occur is very different in the somite position in the developing chick embryo, and then their downstream target genes are very different. So if I was to artificially express TBX5 in a hind region, I would get a wing phenotype, or at least some wing element phenotypes. If I was to express TBX4 in these somites, I would get a developing hind limb in the wrong place. So Hox gene mutations do not always result in homeotic transformation. And the reason is so for instance, if I had a Hox gene here, there's some redundancy, there are duplications, and so I might not get as clear a homeotic mutation. But if I look at these downstream genes, right, Hox genes control where these individual TBX5 versus TBX4 are going to be expressed. And so if I mutate them because of redundancy, I don't get as distinct a homeotic mutation in the vertebrates. But if I ectopically express these downstream genes, I can get a very clear and clean homeotic mutation where hind limb might turn into a forelimb. Now, in a later discussion, we're going to look at mammals and their cervical vertebra. We're also going to look at snakes as a interesting evolutionary kind of a side pathway. How do we get a snake? Because snakes are kind of cool. They don't show any pectoral girdle. They don't show any pelvic girdle. Well, actually, there's a hint of some pelvic girdle in a very few snakes, but most of them show no remnant at all. But their ancestors, if we look at their phylogenetic placement, right, snakes are a subgroup actually of the lizards, and of course they are closely related to all of the other tetrapod reptiles like birds, like crocodilians, a lot of extinct ones, but they're very clearly remnants of and relatives of those four-legged vertebrates, and yet snakes have lost their legs. So how do we lose the definition of a pectoral girdle where forelimbs are going to grow? How do we lose the definition of a pelvic girdle? We'll look at them as a model of a novel evolutionary change in this basic ground plan of the tetrapod vertebrates. But that's the subject for a later lecture.